Okay, good morning all. Can you hear me okay? Somebody give me a thumbs up. Just going to hang on for another minute. People are joining quite rapidly now. Okay, we'll just give it one more minute and then we will start. Okay, right. Well, good afternoon now, everyone. We'll get started. I think there's plenty of people there. Some others will be joining shortly, I'm sure. So welcome once again to the next in the series of the ProLab scientific webinars. As you can see, very straightforward title for us this time, the impact and laboratory diagnosis of pathogenic E. coli. We chose to do this one because there has been quite a lot of interest, a lot of research and a lot of trials running following recent outbreaks. I'm not going to say anything else about that because we have two speakers with us today. Dr. Neil Bentley, our scientific officer here, who you all know. He's just going to run us through the general impact of laboratory diagnosis. And then we are delighted to have Leslie Allison with us, the Deputy Director, Principal Scientist from the Scottish E. coli Reference Centre, who is going to run through all kinds of exciting things that have been going on over the last months up in Scotland with outbreaks, etc. Okay, and then just my last slide, please, Kerry, there. As you can see, these are never commercial, okay, but we are going to be sharing all of the presentations afterwards, and we're also going to be sharing an electronic copy of a sort of brochure booklet that was put together by Neil and myself and some other people recently used at the Scottish Microbiology Association meeting in Crieff. Okay, and we will also be dropping into the chat and providing details afterwards for the products that are available from ourselves, the latex kits that have been used in the trials, and the PCR options that we have. The book, as I said, the NECWAS scheme, I would be interested to know, that has been released today. Now, the last information that we had, and if anybody, please jump in and add to this if you can, was that the scheme was going to be released today. People who applied to join the scheme, it's non-scoring, were told that they would be informed if they'd been accepted. But I don't think anybody's been informed, but it's due out today. Has anybody, just to sort of stick a hand up, actually received any samples or notification for that? No? Oh, okay. Well, I'm sure we will find out afterwards. And I just also wanted to thank E&O Laboratories as well for their support in helping us with the trial, which is taking place up in the Scottish Reference Centre. And I know some of the guys are online here this afternoon. So thank you for that. Oh, a hand just went up. Rupert, you've actually received that. That's good. Okay, excellent. I'll send you a box of Microbank. You can split it up. We can all have a go. Sorry. Okay, right. So without further ado, then I will hand over. I will stop my sharing here and hopefully not stop the entire meeting. If I do, Kerry will run down the corridor and sort it out. And I will hand over to Neil.
Hi, hi everyone. Can everybody see my screen? Can somebody say yes or
Yes, thanks.
oh, thanks. Okay, right. So just to thanks for that introduction, Mark. And if you have any questions, can you put them in the chat and we'll try and have a look as we go along and hopefully be able to answer them at the end. And if there are any questions that we can't answer, we'll make sure we get answers to you afterwards. So I'll try and just move my screen over and just go through. So Mark's already told us what the title is. So let me. Why isn't that moving along? All right, okay, I think it's moved along now. Yes, I'm going to briefly go through the history of E. coli, just to update us, because I'm sure everybody knows a lot about E. coli, probably a lot more than I do. But I'll just go over a bit of the history, some of the benefits. Not all E. colis are horrid, so we can go through some of the benefits of E. coli, and then go on to the pathogenic E. coli. Talk a little bit about STEC, which I'm sure you all know about, and some of the common symptoms that E. coli can cause. Some of the identification of E. coli, and particularly when we come on to the pathogenic E. coli and how we identify them and do strain identifications. And then I'm going to pass over to somebody that actually knows a lot more about E. coli than I certainly do. So here we go. So E. coli was first seen or identified by the pediatrician Theodore Escherich, and was originally called Bacterium coli commune, or later Bacillus coli. It was a lot easier back in those days. So sometimes think that we should call everything just Bacillus coli. It'd make life a lot easier. Um, Obviously, the Escherichia part of the name honors Theodore for identifying the organism, and the coli refers to its origin within the colon. And interestingly, it was initially classified as a harmless commensal and not was not thought to cause any pathogenic disease whatsoever. But we know different, obviously, now. And the first outbreak that of any significance was a food poisoning outbreak in Wishaw in Scotland, which I believe young Leslie at the beginning of her career was certainly involved with, and maybe she can talk a little bit about that later. And in 2011, we probably all remember the outbreak in Germany, which killed 53 people. Um, E. coli, as people probably know, is one of the most studied organisms in microbiology, and I can come on to that on the next slide. And it's extensively used in the study for microbiology and genomics. So the reason being that it's very fast replicator, and we know all about the genome because it was one of the first organisms where the genome was mapped. So it's involved in gut health microbiology. Certainly, you'd want to see it in a gut, and if it's not there, you've got serious problems. So it's associated with gut health. It's also involved with synthesis of vitamins, particularly K and B, and it's also an aid to iron absorption. It's used in biotechnology and research. Within genetic engineering, it's used for a whole host of studies, and including transponon mutagenesis, genome-wide association studies, synthetic biology applications, and bioinformatics databases, etc., and functional genomics, and the list goes on and on. How it's used in genomics. As I've sort of indicated, this is a model organism, and it's the go-to organism for when you're thinking about research. It's also used in food production, in the fermentation industry, for things like yogurts and cheeses. And it's also used as an indicator organism with environmental modelling, and it's one of the first organisms that you'd look for in in waters, etc., to see whether there's any fecal contamination. Also used in health in probiotics, particularly if there's C. diff involved, and also vaccine development. And you can also use it to break down products in bioremediation. So now coming on to the pathogenicity of E. coli, as indicated, most strains are harmless in the gut. It's when they get out of the gut and get somewhere else that they tend to cause problems. And these are the extra intestinal or XPEC strains, and any E. coli can cause a problem if it gets into the wrong place. And it's as you know, it's the most common cause of UTI, particularly in females. It's associated with dwelling catheters, can cause a really bad sepsis, and bacteremia, septicemia, and sepsis. It can cause a meningitis, pneumonia, skin, soft tissue, wound infections. I'm teaching you to suck eggs here, but interestingly, if you look at the picture from the WHO, which has literally just been released, it's one of the organisms that they want the vaccine research and development to be escalated to try and find a suitable vaccine. And if you look, it's the first. I don't know whether how big it's on your screen, but it's the first column in the semicircle. Extraintestinal pathogenic E. coli, and if you look, every region within the WHO, within the world, has requested that we produce a vaccine and escalate things there. So it's showing its importance here. Okay, so now going on to some of the strains that cause GI symptoms, and I'm sure you kind of all know this anyway, but just a very, very quick canter through these, because I know Leslie's going to go into details a lot more on this, and particularly some of the non 0157 STECs. So there's ETEC, which is toxin producing strains, and it's commonly associated with travelers' diarrhea. Enteropathogenic, these E. coli adhere to cells and can cause inflammation, and it's particularly associated with infants and immunocompromised people. There's the enteroinvasive strains. They invade and multiply within the intestinal cells and can cause fever and abdominal pain. There's the enteroaggregative, and these can form a biofilm and can cause a chronic type infection. It's associated with children and the immunocompromised, and then the Shiga toxin producing STEC strains, which the majority of this talk is about. Okay, so just very, very briefly, talking about STEC strains, it can cause a severe diarrhea, often with blood. Some of the other ones do have blood in the diarrhea as well, so it's important to be able to identify these organisms correctly. It's associated with undercooked meats, vegetables, salad, unpasteurized dairy, etc., and contaminated water. It can go on to cause a hemolytic uremic syndrome, which I'm sure you're all aware of. It was thought that E. coli 0157 was the predominant strain, and it certainly was. But now there's other strains that are coming up, and there's what they call the non-0157 STEC strains, and there's certainly globally the big six, they call it, and those strains are associated with disease. And as Leslie will tell you, it's more common to see one of these other strains rather than 0157. These big six, they can be quite difficult to diagnose in the routine lab, and certainly that's why we're doing the research here, to try and sort of move that along and make that an easier diagnosis. Okay, so going back to E. coli overall, what sort of pathology does it cause? Obviously, the different strains are dependent, different pathologies are dependent on the strain and the infection associated. So it can cause a UTI, can cause sepsis, a meningitis, pneumonia. The GI symptoms include abdominal cramps, diarrhea, sometimes with blood, nausea and vomiting, fever, and as I've said, hemolytic uremic syndrome. Now on to identification. Very, very briefly, I'm not going to go into genus identification. It's very easy. You do it every day in your lab on numerous isolates. So I'm certainly not going to tell you how to load a moldy or do an API or even do sugar reactions for us old enough to remember sugar reactions. But specific strain culture and identification can be difficult, and that's particularly why we now need chromogenic media. And I know E&O are involved today, and they've certainly got a fantastic agar that can be used. But we do need to be able to differentiate these. We need to be looking at all the different antigens associated with this, the cell wall and the flagella antigens of these bugs. So the O, H, and K antigens are really important. There are molecular methods, but obviously with molecular methods, you'll only see what you're looking for. So the molecular methods are more associated with the reference laboratories, and we should be thinking about how we can actually diagnose this in the regional labs away from the reference lab, perhaps not to the molecular level, but certainly to serotyping levels, because I do know that both certainly the Scottish and the English reference labs are overrun with the amount of samples that they're receiving. So now I'm going to shut up and pass over to Dr. Leslie Allison, who will tell you everything you need to know about non-0157. So Alison, so Leslie, I'll stop sharing now, and hopefully you can take over.
Yeah, thanks very much, Neil. Great as always. Just wait while Leslie gets a screen shared there. UK Equus did confirm in the chat, I'm sorry everybody saw, so many thanks for that, that these samples have been sent this morning, so they will be received by the labs this week.
Right, sorry there. Let's try again. I was having problems sharing.
Okay, no problem.
There we go. Let's try this. Screen. Okay, can everyone see this? And I'll.
Yeah.
Yep. Should
Perfect.
be stop. Yeah,
Okay, there we go. Let's come out of this. Goodness, right. I'm hoping that's us.
Yeah, that's fine.
Right. Fabulous. Right. So thanks so much, Mark and colleagues, for inviting us to come and talk today. Neil, thanks for that introduction. I'm just really going to go through, this is very much a setting the scene presentation. I'm going to talk a little bit about who we are, what our methodology is, our current challenges perhaps, and a little bit about some preliminary work we've been doing on a culture method to try and help us in the lab to isolate non-0157S tech, with also a little bit about future work we are currently undertaking. So for those of you who are not familiar with us, we are the Scottish E. coli 0157S Tech Reference Lab. We were initially set up in Scotland in 1993 in Aberdeen, and then in 2000 we moved to the Royal Infirmary of Edinburgh, and that's where we are located just now. Our remit hasn't really changed over the years. We provide identification and typing of E. coli 0157 and other STEC in order to guide the clinical management of individual patients and also to supply detailed information for health protection purposes. Quite a large part of our workload is fecal screening of high-risk, locally negative samples, and I'll talk a little bit more about that. And a lot of our workload includes the isolation stage from these PCR positive samples, and that is not straightforward. And once we have an isolate, we do whole genome sequencing, and we obviously play a role there in outbreak detection. And just to say, at this stage, GI PCR or enteric PCR isn't fully rolled out to all the Scottish diagnostic labs yet. I think there are 3 Scottish labs that we're aware of that have gone live, and they're all using slightly different platforms. So this is our Scottish Health Protection Network current guidance. It's 2018 edition. It's currently under review, and we're hoping the new version will be rolled out by the end of the year. And this really defines what we mean by high-risk samples. So these are currently cases of suspected HUS or cases of bloody diarrhea that are culture negative locally, symptomatic contacts of these cases, or any outbreak associated case, and that could be symptomatic or asymptomatic that has tested negative locally. And these are what are classed as high risk that then should be sent to us at the reference lab for further testing. So just to give you an indication of what we are receiving in the lab from a sample perspective, this graph shows you our kind of samples submitted since the lab opened in Edinburgh in 2000. And what you can see here is a steady backbone of isolate submissions, so as evidenced by the blue dots and the blue line. So that's a fairly steady backbone here. And I would say almost without exception, these isolates are E. coli 0157. But you can see the ongoing increase year on year in fecal sample submission to us. The increase has really been for a number of reasons. Certainly the guidance and the various iterations of the Health Protection Network guidance advising labs to send samples to us, and perhaps a slight delay while labs formalise their SOPs to send us the samples. So changes in guidance has meant over the years more samples have been sent in. We had a little bit of a lag for various reasons, not least COVID, although COVID didn't impact us on too much. We were still getting a sort of similar level of positivity. Bear in mind, these aren't positive samples, this is just sample submission. And then we saw a very large peak in 2022. I'll come to that a little bit later. Last year we were down a little bit, but our current figures for the past 7 months show that we are currently up by 6%. So still that upward trajectory continues. The landscape of STEC seems to be changing. We've noticed it for a number of years in that there's been a slight decrease in E. coli 0157 and an increase in non-0157 STEC. But last year was the first year that non-0157 STEC as a group overtook E. coli 0157. And you can see that peak here in blue. But that's very much explained by what we call our Summer of Outbreaks 2022, where we had overlapping consecutive outbreaks of STEC. And while the case numbers don't look particularly high, there was still a very large burden of fecal samples submitted. And these outbreaks, we'd noticed really for the first time, were actually quite complicated. We were finding, multiple strains within an outbreak. We found patients who had dual infections, dual STEC infections. And we even had a couple of cases who we were clearing for one S-Tech and just about to clear them, and suddenly we were started to detect a different S-Tech, and we had to start the clearance process all over again. So it was an incredibly challenging time, yeah, submission continues. So what are we actually isolating? This graph shows the strains that we are isolating and have sequenced, in particular the non-O157, and this is just 7 months of data from April this year to the end of October. This doesn't include the PCR positive fecal samples where we've not been able to isolate an organism. And you can see quite clearly that for most of these serotypes, we're only isolating one. So it's only seen on one occasion only. It's been sporadic infection. Again, just 2, 3, or 4. I mean, it's likely these have been family clusters. But what is striking is, this distribution here of the top 3, O145H28, O26H11, and O103H2. These are by far the top serotypes we've seen this past year. But in saying that, they've also been associated with outbreaks of infection. And just to crunch the figures a little bit more, for the past 7 months, those top 3 serotypes we've seen in 70% of all the serotypes we've managed to isolate. Last year, that figure for those top 3 was 60%, and the previous year it was 42%. So how do we go about isolating these at the reference lab? Well, when the fecal sample arrives, we enrich overnight in TSB, and then we do a manual extraction, and we use an in-house PCR. We're looking to detect the RFB 0157 gene, the Shiga toxin 1 and Shiga toxin 2 genes, including all variants, and that also includes Shiga toxin 2F. And we also include an internal amplification control, can't say that word, internal amplification control in each reaction. And just to say at this point, PCR detection is by far the easiest part of this process. The isolation stage is not straightforward. We can get a feel for what organisms might be there by looking at our genes detected. So clearly, if RFB0157 is present, that's a very strong indication that E. coli 0157 is the organism we're looking for. If RFB is not present and just Shiga toxin genes, then we're potentially looking for a non-O157 S-Tech. We're aware that for those using GI-PCR, not all platforms detect RFB, so you won't have that sort of division. And also we need to be mindful of a dual infection. So if all 3 genes are present, we've not to assume that all 3 genes are from the one strain. And we have seen a number of dual and sometimes triple infections over the years. So on the right-hand side, this is our process for isolation. So we take the PCR positive broth. If it's an incredibly strong CT, we do direct plate, and we may be fortunate and able to isolate, but invariably what we will do is do a serial dilution of the positive broth and plate out the lower dilutions and spread plate these for single colonies onto a SMAC and a CT SMAC. And then the plates are scrutinised for different colony types, be it different colours, different morphologies, and we take, on the first pass, we'll take 20 colonies for PCR. We call it our 20-pick PCR. That will be 18 picks, and then we take a sweep from the SMAC plate and a sweep from the CT SMAC plate. And just to say that some non-O157 serotypes are sensitive to fixed and tellurite, so sometimes we don't see any growth on the CT SMAC, but we do see on the SMAC, which is why we plate onto both. Obviously, there's challenge to current culture methods, and the protocols in the UK, and actually surveillance as well, has been very much geared towards E. coli 0157. And on the left here, you can see very clearly E. coli 0157 is a non-sorbitol fermenter, and it's a lovely biochemical quirk, which means that the colonies are a milky colour when they're going on SMAC or CT, and you can very easily pinpoint the colony and do an 0157 latex, and then you know that's what you have. In contrast, the non-0157 S-Tech are invariably sorbitol fermenter, although some are non-sorbitol fermenters, and they'll look like any other E. coli, commensal E. coli on a culture plate. And bearing in mind, you actually don't know what serotype generally you're looking for at this stage. So you can't just go in and do latex here. So it's a real challenge for culture. So we've been looking at chromogenic agar, and we've been looking at ColorX, S-Tech, and I know others have as well, and I know others have already published on this, so they'll probably know much more than we do. But we've been using this as an aid to isolation in parallel with our current method. And what we're finding is S-Tech grow as mauve colonies almost without exception. Commensal E. coli and other enteric bacteria are mainly blue-green or colourless, but you can see here there's different variations of colour that we have come across whilst looking at this. And one of my colleagues, Mary Mitchell, presented at the SMA meeting in Crieff last month. And I think her slides may be available as Mark alluded to earlier on the webpage. This is one of her slides showing that I think we looked at 200 or so positive samples, and of those where we managed to isolate an S-Tech, 70% of these were isolated from the ColorX agar plate, and only 30% not. So whilst S-Tech grows mauve colonies, not all mauve colonies are S-Tech. The other slight issue that you do have to bear in mind is the ColorX S-Tech plates also include cafixime and tellurite, so there will be strains that won't grow. They will be if they are resistant to tellurite and cafixime. And some E. coli serotypes grow better than others on the chromogenic agar. And we know it's very good for E. coli 0157H7, O26, H11, O145, H28, and most O103H2, which is why it's been quite helpful for us during the recent outbreaks. But it's all very well growing on the plates. What do you then do with them when you have a mauve colony? Yes, you can PCR them if you have PCR capabilities, but if you don't, or in addition to that, there is now the non-0157 latex kit. We've been using this in anger during the recent outbreaks, and it's been super helpful. You'll all be familiar with the 0157 latex kit, and this is exactly the same, but the kit includes latex for the top 6 serogroups that Neil alluded to earlier. Important to say that these include O26, O103 and O145. So it captures strains that we've been seeing a lot of outbreaks with. And also these are the serogroups that tend to be associated with severe disease, because we know that these carry the Shiga toxin variant 2A in conjunction with EAE. So it's a great steer and a great help for us. So I'm not going to show you any data. We're actually still teasing out the data from the outbreaks. We keep hoping for break-in outbreaks, and it's not happened yet. There's a lot of data there, but overall, what we are finding is when this is included as part of our protocol, we're not having to spend time doing lots of 20 picks if we don't get PCR positive reactions on the first 20 picks. What we would sometimes do is repeat that again, Or alternatively, we might pool picks. So we might take 5 colonies from a plate into one PCR reaction and do that 16 times. And it's all time, but we've shown during the outbreak that we've sometimes just been able to go straight to the mauve colony on a plate and it's latex positive. And I can't begin to tell you how much that streamlined our workload, because at points we've been drowning in a sea of colony picks when we have multiple positives going through the lab. And the benefit of having that positive colony earlier is we can have much faster whole genome sequencing results. We need an isolate for whole genome sequencing. So this has really let us speed up that protocol. And obviously, you know, we are then able to pool cases into outbreak investigations. It's obviously been super helpful for us during the recent outbreaks where we've known the serotype. So as I say, we've been using it as an aid to isolation, but it could have other uses when screening family clusters, for example. So if you know a particular family member has a 103, you could use that to screen the extended family. So it could be used sort of as direct plating, and it may have a role in clearance, just like you use 0157 latex to clear locally. The same could be done here if you know that the patient is infected with a serogroup that's in the kit that there is a latex for. In terms of future work, Mark alluded to this, we've just started a field validation of the non-0157 latex kit with ColorX, SMAC and CT SMAC. As a department, we've signed up to the UK NECLAS Educational EQA, so we're looking forward to receiving those samples through hopefully this week. And what we're doing now is teasing through a lot of data, and I'm now writing a business case to try and include the chromogenic agar and the non-0157 latex. Like everyone else, we can't just make a decision to change up what we do in the lab. We need to submit this to our funders, and it goes through various levels of scrutiny. So it won't be straightforward, but I really do feel we can demonstrate how helpful it's been over the past year. So fingers crossed on that front. And just finally, thank you to my lab staff. They are the greatest. They have been incredibly busy over the past couple of years. I keep telling them Christmas period should be quiet, but it certainly wasn't last year. Fingers crossed going forward this year. A huge thanks to our NHS diagnostic labs who send us samples with lots of information attached. Thanks to ProLab and E&O, they have been very responsive to our orders, particularly during the outbreaks when we've really relied on them to get through, and our funders, NHS Scotland, NSD. I just also say, as a reference lab, we are impartial. You know, we don't promote one particular company or a product over another. I think it's just important to say that. And I am happy to take questions. Thank you very much.
Okay, thank you, Leslie. Excellent, absolutely excellent, very informative. Thoroughly enjoyed that. So I'm going to hand over, I think, to Neil to sort of field questions. You can either drop them into the chat, or if you want to do traditional pop your hand up, we can take you off speaker. As I said, we are going to be releasing everything quite quickly this afternoon. And if there is anybody that's intending to start and wondering how they're going to do it, just let us know if you need one of the non-0157 latex kits, which covers the Super 6, and we can sort that out for you.
Thanks.
Okay.
Thanks, at the moment, I can't see anything in the chat, but I'll.
Georgina.
Oh, Georgina, yeah.
Okay, how do we take Georgina off mute, Kerry?
Hi.
Okay.
I'm interested in how specific the chromogenic agar is now, because we trialed it in our lab a couple of years ago, and it gave us so many false positives that we had to send to the reference lab for confirmation. And they all came back negative as being a toxin producer. We stopped using it.
Okay. Alison, do you want to handle that one, or shall we ask Richard to come in?
Um,
Or both?
why don't we go to Richard? I mean, in our hands, it's performed really well. But
Yeah.
maybe go to Richard first.
Yeah, we used it at the Georgina. We used it at the UK HSA labs as well, where we did some trials that were centered in Bristol, and then the 5 UK HSA sites. I'm not sure if anybody's online from there, but Richard, if you could come off mute and be put on the spot, that would be great.
Yeah, well, I'm based at the Bristol lab, and
Oh, right.
I think we ended up
Okay.
doing
Ooh.
2 trials, and this is about the first one. I wasn't involved in the second one, but I felt rather guilty because I was the one who said, "Oh, let's try this out," and it was such a disaster first time round that we stopped using it.
Maybe I can come in while Richard's still trying to come off mute. I know we are having to be particularly mindful of sort of sugar toxin negative strains. So sugar toxin negative O26, 0145, because chromogenic, we're picking them up, but then they're not testing positive as latex. So that's really been the one kind of slight issue we've been seeing. Or perhaps it's just because we have been so skewed towards 0145, 026 that we've maybe just not come across the issues that you've seen, Georgina. But I'd be quite interested to talk a little bit more, maybe offline, about that and about your experiences. And maybe we'll pick that up a bit more when we go forward and do
I think we can,
the trial.
Well, this was during the time when I don't think the non-0157 latex kit was available. So all we could do was go by colour of the colony on the agar plate. And we came up with so many potential positives that it was, it was very difficult to report. And then, of course, they all came back from the reference lab as they were all negative.
That's Neil here. Richard can't get off mute, so I don't know whether Kerry can. But in the meantime, I can. Oh, Richard, you're on.
Is that me? Here we go. Can you hear me now? Yeah.
Yeah, we can. Yeah.
Yeah. Sorry, Georgina. Which one are you from?
The Bristol UK HSA lab. I wasn't involved in the later trial, so I don't know what happened then. But
the later trial was using the kit as well, Georgina. So anyway, but we can take that one later. Sorry, Richard. Go ahead, mate.
Yeah, no, that's fine. Yeah, I agree with Leslie. I mean, obviously we've sent some samples out to Leslie, and we've not heard any issues of the negative. So yeah, it'd be good to kind of speak maybe offline to find out what issues you had.
Yeah, maybe I can come in a little bit again here. I guess this comes into the category of, we know all S-Tech are mauve colonies, but not all mauve are S-Tech. And I guess if you don't have the latex, then it's maybe the mauve ones that you're talking about that were the ones flagging as then coming back from the reference lab as negative.
I think that's absolutely right, and I think what we've got to remember here is as with lots of things in microbiology, it's a screen, it's an aid to diagnosis, using one in isolation, a chromogenic media in isolation without a latex perhaps isn't the way to go. Perhaps you need a combination, hence the reason that Leslie's putting a business case together to try and bring this in as a front line. So I think we all understand what screening means, and we will always miss things on a screen or even screen things that turn out not to be sort of pathogenic. However, if we get this right, what it will do, it will allow the reference labs to actually concentrate on the weird and wonderful bugs that are changing and allow us to at least screen out some of them. But as I say, used in isolation of just one thing probably isn't the right way to go. And Georgina, I understand that when you first did this, you didn't have access to the latex, so perhaps the second trial might have been more fruitful for you.
Okay, thank you, guys. Ava. We're just trying to get you off mute, Ava. Won't be a second.
If necessary, if you put your if you put, I think Kerry needs to unmute everyone. So if you have got questions, if you pop them in the chat, and I can, because I'm obviously not muted, I can relay the questions over if necessary.
I'm also happy to answer questions if people are struggling with questions, just to reach out with questions, and we can do this after as well. I'm quite happy to do that. Thanks.
Yeah. I mean, any question, you can either pop them into the chat, guys, or you can drop an email into uksupport@pro-lab.co.uk. And certainly when all of the recordings, etc., are released later today, you'll have all the contact details that you need. So are you okay, Ava? Do you want to put something into the chat, or do you want to come back to us later?
I see that Lewis.
Yes, it is. Yeah.
Yes, it will be available later, Lewis.
Yeah. Yeah. Oh, yes, and I did just answer. Okay. Yeah. You didn't miss anything, Lewis. It was only me talking. Just thought I'd put that in there while we're all. So are we, if there are no more questions, I mean, I know we are rapidly approaching 1 o'clock, and I hope that everybody's managed to grab some lunch or is about to. So just in closing, thank you very much, Leslie. An absolute pleasure as always. And Neil, thank you as well. And we'll be in close contact with Alison throughout the trial. And at some point down the line, we'll be able to share all of that wonderful work as well. What does that mean, Caroline? Okay, I don't understand that message. Sorry. Am I missing something there, Neil?
No, I don't know what that.
Okay, yeah. All right then. Okay, so we'll call it an afternoon there, guys, and we'll be releasing everything in the next couple of hours. And thanks again to Richard for joining us from E&O. And watch out for the next one. We will try and get one done. I think it's going to be New Year now, unless we can get something put together before Christmas. But I think New Year's always quite good fun with all the various bugs and things that'll be flying around at Christmas. Okay, and thank you all for your very positive comments there. Okay, guys, we'll catch up very soon.